Good evening, Mr. Parisas. Today I'm going to be explaining the process of protein synthesis through transcription and translation. Transcription is defined as the process where a cell makes an RNA copy of a section of its DNA, and translation is the process where the genetic code carried by a messenger RNA into bracket mRNA molecule is used to build a specific protein. It is the second major step in gene expression following transcription. So we're going to begin with transcription. Transcription is defined as the process by which DNA gets converted into mRNA, also known as messenger RNA. I know I defined it already, but twice the better. So these are the steps in transcription. There's initiation, there's elongation. And initiation is the process. In initiation, the RNA polymerase comes and connects to the DNA in the nucleus, which is the control center of a eukaryotic cell where the DNA is located. So on our diagram above, we could see the initiation and how the RNA polymerase is connected to the mRNA and the DNA structure. Yes, and in step 2 of initiation, RNA makes the two DNA strands to unzip. And unzip refers to the process of unwinding and separating the two strands of the DNA double helix. As you could see how the in the diagram the strands have been unzipped. I don't know if you could see it, but so if you look closely, you could see that the DNA have been unzipped. And the second step is elongation. Elongation is when RNA keeps reading the genetic code and the mRNA strand gets longer. During elongation, the RNA polymerase begins to add nucleotides to the growing mRNA strand, which is defined as a single-stranded molecule that carries the genetic information from the DNA in the cell's nucleus to the cytoplasm. Thus, RNA polymerase starts making mRNA from 5 to the 3 end. In the previous diagram, there was the 5 and the 3 end on the DNA structure. Yes, however, RNA polymerase reads the DNA strand in the 3 to 5 direction on the template strand. The non-template strand is also known as the codon strand, which is not used because its sequence matches up with RNA, except the fact that uracil is found in RNA and thymine is found in the DNA. So the final step we're going to talk about is termination. Yep. So termination is when the RNA polymerase molecule, the mRNA strand, all separate from the DNA template strand. So I don't know why the edit is still going on. It's supposed to end now, but let's ignore that. In this step, the polymerase enzyme caps the 3 ends of the mRNA strand, and this is known as the poly A tail. At the end of the termination step during transcription, DNA has been used to create a pre-mRNA strand. This strand has something known as introns and extrons. Once the RNA finishes reading, it disconnects and termination has been completed. And now we have a completed mRNA strand. So if you look in the diagram, you could see how the strand has been disconnected from the I indicated on the diagram that the mRNA strand has been disconnected and termination has happened. And once termination happens, translation is next. Now that we talked about the first half of the polypeptide synthesis, we move on to the second half, which is known as translation. Yay! Translation is the process of taking the information stored on an mRNA strand and using it to construct a protein. Once the mRNA strand is synthesized in the nucleus, it leaves the nucleus and enters the cytosol where enters once the mRNA strand is synthesized in the nucleus. It leaves the nucleus and enters the cytosol, where it attaches to a free ribosome. So I'm gonna brief everything about translation in these few sentences that are gonna come out. Within the ribosome, the mRNA is going to interact with a tRNA molecule, which is a small non-codon RNA molecule that serves as a crucial adapter in protein synthesis. The sequence of the nucleotides on the mRNA strand are separated in sets of 3, and each set of 3 nucleotides represents a codon. A codon is basically a sequence of 3 nucleotides which together form a unit of genetic code in a DNA or RNA molecule. So on the diagram above, you could see that I pointed out which 3 codons were, which is the codon AGA, and I also pointed out the mRNA and the tRNA and the ribosome and the strand, which the codons are attached to. And yes, the first 3 sets of nucleotides matches up with another set of 3 nucleotides known as anticodons. An anticodon or an anticodon, the first set of 3 nucleotides matches up with another set of 3 nucleotides known as anticodons. An anticodon is a sequence of 3 nucleotides located on the transfer RNA molecule known as codon, and each codon matches up with a specific amino acid. Amino acids are defined as the fundamental organic molecules that serve as the building blocks of protein, and that is basically the brief up of initiation. Now, elongation in translation is similar to elongation in transcription. So during elongation, the tRNA molecule enters the ribosome through the A site and lands on the P site. The amino acid gets attached to it and then moves to the E site. It loses the amino acid from the P site, and due to multiple tRNA in the ribosome, all detached amino acids creates a chain which is called the polypeptide chain. The polypeptide chain will therefore extend out to the ribosome and then the tRNA. Eventually, the tRNA leaves the E site where the tRNA is removed. The final step is indeed termination. Termination begins when the stop codon is read. The stop codons get read at the A site, and that's the factor that realizes the polypeptide molecule. The protein therefore leaves the ribosome where it will eventually go to the Golgi body for further processing. The RNA moves across from left to right and starts the mRNA change, while mRNA strand copied code becomes complementary. And yeah. So before I wrap up, I want to show you how the amino acids how it looks when it comes into a chain, and then show you how a polypeptide chain looks once it's folded. Thank you so much, Mr. Parisas, for listening to my little presentation. I know it's not the best, but I tried to do as much as I could, and this is truly my best, and I suck at editing. But thank you so much.